Good morning, all. We are now starting with the webinar. I, Ms. Sharayu More, on behalf of Department of Microbiology and Biotechnology of Patkar-Varde College, welcome you all for one-day RUSA-sponsored national webinar, Biofacet. It gives me immense pleasure to welcome our guest speaker of the day, Dr. Abhijit K. I will now give a brief introduction of our college. Chikisak Samuha's Patkar-Varde College was established in 1964 in Mumbai's western suburb. Our college is an autonomous college affiliated to University of Mumbai and been re-accredited with A+ grade by NAC third cycle with an institutional score of 3.53. We have also received Best College Award in 2016-17 from University of Mumbai and are a DBT Star College awardee. We have also obtained ISO 9001:2015 certification. Since its establishment, it has constantly worked towards student and society welfare by bettering its infrastructure. The college has grown over last 6 decades and in keeping with the spirit of learning and growing through criticism, we have set standard high, working continuously towards advanced goal. Our college offers a wide range of undergraduate, postgraduate and PhD programs. In addition to the traditional programs, the college also offers need-based programs like BSc in biotechnology, microbiology, aviation, computer science, information technology. BMS, BCom in banking and insurance, BCom in accounting and finance, MCom in advanced accountancy, MCom in banking and finance, and MCom in business management. A brief introduction about our department. Department of Microbiology and Biotechnology was established in the year 2006. The department has been organizing Biofacet every year. Started in academic year 2012-13, Biofacet is a stride taken by the department to provide a platform to scholars, teachers, and experts from various fields of science and technology for academic interaction, elevation, and enrichment. It has also taken initiatives in helping the students in extending their mind in diverse fields available in biological science. This year, we have organized two national webinars on fungal genomics and its implication and advances in cancer research. And this is the third webinar in the series. We are extremely glad to organize this again, and this would have not been possible without your support and participation. Thank you, everyone. Today's session is on sustainable agriculture through plant breeding. As we all know, that plants are important to mankind from the earliest study of life process, and they are also central to scientific study. Research on plant enriches our intellectual life and add knowledge to other life processes. Our focus today is on molecular markers, concept and its application in crop improvement. Dr. Abhijit KP will guide us through this subject, offering insights on how molecular markers are shaping the landscape of sustainable agriculture. With a great honor, I introduce today's speaker, Dr. Abhijit KP, a distinguished scientist in genetics and plant breeding currently contributing groundbreaking research at ICAR IARI Assam. Holding a PhD from ICAR IARI New Delhi, Dr. Abhijit specializes in basmati rice heteroic grouping and has made notable strides in magazine biofortification program. As a scientist, he actively engages in international collaboration, demonstrated by his professional attachment training at International Rice Research Institute, focusing on halotype-based breeding of rice improvement. Dr. Abhijit's impactful publication on low-phytate genotype showcased its commitment to addressing global challenges in agriculture. Join me in welcoming Dr. Abhijit KP, a visionary scientist whose concise and influential contributions are driving positive transformations in the realm of genetics and plant breeding. It is indeed our pleasure to have such an eminent speaker with us today. Before we begin the webinar, I request all the participants to be kindly patient if any technical problem arises during this webinar. We will try to resolve all the technical glitches. Kindly post your questions in the chat box and they will be answered at the end of the session. We are sure that this webinar will certainly complement your learning objectives. Now, I would request Abhijay sir to address the webinar.
Hi everyone. I hope you are all doing well. And thank you for a kind invitation. Okay, just before starting, I would like to check that my audio is visible. Sorry, audible, and my video is visible.
Yes, sir, you are audible.
Okay, great. So I am anticipating some technical issues with my internet connection. So probably some of my slides may not move. So just let me know in case any technical glitches occur. Okay.
Yes.
Yeah. So again, I'm really being grateful to get this opportunity to talk with you all and regarding. I hope I will try to bring some new thing too in front of you all. And may I share my screen? Okay. So my screen is visible in the full screen.
Yes, sir, it's visible.
Yeah. Okay. Okay. Right. So today we talking about molecular markers concept and its application in crop improvement. And yeah, I understand that the audience is little have different disciplines. So not everyone of will have very basic, very thorough basic concept about either about markers or about crop breeding. So my intention is to have a broader knowledge rather than going to very specific aspects of each of these. So the presentation will be mostly about how crop breeding can bring changes in ensuring the food security, and also the importance of the molecular markers playing in having better breeding potential. So with this, I would like to start my presentation. And okay, yeah, before starting it, I would like to have a brief outline about the evolution of breeding of field crops in India. And yeah, I understand that it is your institute is a non-agriculture based institute. So this is very basic aspects of crop breeding. So we have around in India has seen, if you see the if you talk about the crop breeding, India has seen 6 different phases of, even the world has seen the 6 different phases of crop breeding. And the first phase is introduction. Introduction is bringing the genetic material from outside to a particular country or a region from whether that crop or plant which never has been natively grown. And thereby you can bring a lot of changes. So some of the notable introductions are in 1960s. So in case of rice, we have brought better yielding rice varieties from Philippines. And in case of wheat, we have brought better yielding wheat varieties from Mexico. That has played a significant role in ensuring food security at that point of time. And yeah, other phase is selection. So in self-pollinated crops, we go with the pure line selection, whereas in cross-pollinated crops, our idea is go for mass selection. And the next phase was hybridization and selection. And here we take two parents, two plants, we cross them, and
Hello,
yeah, hello.
sir, your screen is not visible
Ah,
or not moving. Slides are not moving.
Okay, okay, okay. Right.
But it was visible here, so
Oh, it was visible.
yes,
Okay. I will try to share it again.
yes,
Yeah. Is it visible? It
yes,
is must
yes. It is
it
visible.
must be on evolution of breeding field crops in India.
Yes, right, sir. It's visible.
Okay. 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 So yeah, the next phase was mutation breeding. And mutation breeding, you are using physical or chemical mutagens and causing, introducing some mutation in the plants, and thereby you are changing the traits of your interest into, the better yielding or the way you want to change the traits. And this phase was made a tremendous improvement, change in crop breeding in India. It has, we have released a lot of varieties just through mutation breeding alone. And the next phase was in hybrid breeding. So in cross-pollinated crops, the hybrid breeding was playing a very big role since from 1920s. But in case of self-pollinated crops, it was relatively a new concept. So hybrid breeding was, again, it has a potential to increase the crop yield. And we have been practicing this since 1970. And yeah, then the next phase came for molecular breeding. So molecular breeding, yeah. So the initial application of molecular markers were majorly on humans. So saying the potential it has. So the plant scientists took thought about why can't we apply this in our programs, plant breeding programs, and then we have incorporated, starting with RFLP, we have incorporated these markers into our breeding programs. And molecular breeding programs are still relevant today. And in 2006, if you see the post 2010, most of the crops are getting sequenced and already sequenced. So now the era of genomic assisted breeding. So we will have a brief outline about all those things in the coming slides. And again, the plant breeding, those who have a very preliminary idea about the plant breeding is it's both art and science. How a breeder select a plant or genetically modify traits of a plant or the whole plant as such to bring and improve the genetic makeup so that the plant can be used for the better, the plant can be used for the human benefit. And conventionally, for example, how we breed is we will raise our experimental plots a population of plants, okay, and these plants uh, we will grow the plants once. Okay, again, I'm sorry for interrupting. Is it again? Is it visible? The animations are visible. Yeah. Okay. Thank you. So once we grow these plants in the field, then suppose at some point of time, some disease has occurred in the plant, some disease infection occurred. And yeah, not every plants will survive within the disease infection. Only some plants will be wilting and some plants will be surviving. So what the breeder does is, he will carefully select those plants which are surviving, right? Because the plants which are already been infected or already undergone the disease, already, already been wilted with the disease, there is no use of selecting those plants, right? So we will be selecting those plants which are resistant. And the next season, we will sow the seeds of these individual plants. Then, for example, we have selected here 3 plants. Then in case if we sow the seeds of these 3 plants, again we will get 3 plants in the, if you're selecting a single seed, suppose you get again 3 plants. Now when you grow again, the disease has occurred at some point of time, then what will happen? And we assume that since these plants have been already, we have tagged them as resistant in the previous generation, we are expecting they should be again resistant, right? But it may not be the case. Out of 3, maybe 1 survive. Again, the 2 may get disease. But how it can come? We have selected those plants as resistant plants, but the next season you find the same resistant plants showing such stability. How it's possible? So it can be possible due to different reasons. It can be possible due to some errors in the selection. It can be possible due to whether the enough inoculum of the pathogen was not present to transfer the disease into every plant. Maybe the or other reasons can be, maybe the environmental conditions was not congenial to transfer, like spread the disease uniformly across the population. So due to all these reasons, your selection failed now. Okay, even though you have selected 3 plants, now you are left with only 1 plant, right? So to overcome all this problem, again, here you are selecting, not in the initial stage. You want to wait till the disease occur, and then only you will be able to select the plants. Now the problem here is, now to address all these problems, uh, the the scientists have been taught, and that's how the concept of molecular markers came into the picture as a selection tool. So we will briefly see what is molecular markers. Okay, so the genetic markers. So the genetic markers can be broadly defined that a trait that is polymorphic and easily and reliably identified and can be readily followed in a segregating generations. So segregating generations are, for example, you. I hope everybody must have undergone the courses in, I think, 12th at least, about the genetics, right? I'm sorry to interrupt, ma'am. The audience, especially the students, may have some understanding about the genetics, or shall I explain from the beginning?
Yes, sir.
Okay, you have understanding, which is really good. So the segregating generations are, for a particular trait, if the 2 parents are different, then when you grow the first generation, second generation, third generation, you find that the trait will be segregating, meaning the trait will show differences in their expressions. Okay, and so coming back to the marker, and the marker should indicate the genotype of the individual exhibiting the trait. So genotype is what indicating the genetic makeup of that particular trait, whether that trait is, like we say, capital A, capital A, capital A, small a, small a, small a. So that is the genotype, and the phenotype is what visible to you, what you can quantify, right? And it should also, you know, so the plant should indicate the genotype of the individual exhibiting the trait. Now coming to the molecular markers, okay, I will give you an analogy here. So suppose you are moving into going to a place where an unknown place, okay, and this is the situation before coming your Google Maps and all. So how do you identify whether you are about to reach the place or you already reached the place? You will look for the milestones, right? There used to be milestones. I don't think nobody cares these days, because now we all have GPS, Google Maps. So if you have a milestone, it meaning that you are about to reach the place. So molecular markers are just like these milestones. Okay. So if the molecular markers are basically identifiable tags. So if suppose you have already been associated or mapped those particular molecular marker with your gene of interest, it meaning that you just have to search that molecular marker. Meaning that if the molecular marker is present, then the gene is in the vicinity of that marker. Suppose if the molecular marker is present in the population, you are assuming that the plant will be co-segregating, the molecular marker will be co-segregating with your trait also. So, your gene also. So you don't have to look for the gene. Instead, you look for the genetic marker. So you assume that your gene also will be present there. So molecular markers are can be broadly classified into 3 types: morphological markers, protein-based markers, and DNA-based markers. Now coming to morphological markers, okay, these are very easy to identify. These are traits which are visible to your naked eyes, and they represent actual phenotypes. Suppose if you see the picture on the right side, there is a leaf tip burning. Okay, this is a wheat plant, and if you see the leaves of this wheat plant, there is the tip is burnt, right? In the leaf tip is burnt. So this particular trait is associated with leaf rust resistance gene. So suppose if you want to select, if you want to select your plants for leaf rust resistance, all you have to do is look for this leaf tip burning, right? So even though you feel like it's so easy to select these kind of traits, but these markers are very few. Okay, not for every trait or every gene these markers are not present, and also they express in a very specific developmental stage. And also, yeah, again, these morphological markers won't be expressing in every environment. It needs certain threshold requirement, and also it can have some negative pleiotropic effect. And it's very difficult to maintain these kind of genetic stocks in a field. So these markers are not at all used widely. Okay, if it's there, it's an added advantage, but a breeder cannot relay these markers all the time. Okay, then came protein-based markers. So these protein-based markers have been, I think, still rarely we use it, but once upon a time we used to use it widely. And these markers are based on variation in protein structure and function. And the good thing is that it can be detected at early stages also. And you need only few amount of tissue, just extract this protein. And again, the problem is like, the protein is the end result. First, if you see that central dog, if you again recall the central dogma of genetics, what it is from DNA, the DNA will transcribe to a mRNA. From there, it translates to a protein, right? So the protein, it vary with the developmental stage, environment, and tissue, and it's very tissue specific. So also, if the proteins can be even multimers, it can be homomultimers, heteromultimers. So in case of heteromultimers, you will get lot of bands. So with those lot of bands and things, you will be very difficult to interpret your results also. So with all these limitations, protein-based markers are not uniformly adopted. So there comes DNA markers. So DNA-based markers represent variation in the DNA sequences of different individuals. Okay. So the advantages of the DNA markers are polymorphism is the different. The polymer, the technique. So I will explain certain technical terms so that you will be able to follow me. So the by polymorphism, what we mean is, for example, you have two individuals. Okay, so the two individuals have different genomic region, or different, the content of the genomic regions are different in two individuals, that we call in a simple terms as polymorphism. Okay, so here DNA markers have that polymorphism distributed over entire genome. Okay, and they present in high marker densities. Also, you can score one marker without affecting other markers independently. And most importantly, these markers show simple Mendelian inheritance. If in a population, if in an F2 population, they will show if one marker is present, you can just trace it as 1:2:1. And they are independent of prevailing environmental condition. Any environmental condition, you can score these markers, and at any developmental stage, because the DNA is same from, even if a plant is at the seedling stage or at maturity level, the content of the genome is same. The DNA sequences are same. It is not going to change. So you can score them at any stage. And also these marker assays are non-destructive. Okay, you just need a leaf tissue or something, any tissue from which you can isolate the DNA and check the markers. You don't have to, destroy the entire plant to get the result, right? Now, if we go for classification of molecular markers, the first generation of markers were based on the chronology of the development. The first generation markers were RFLP and RAPD. So RFLPs were, they have discovered, or they have been introduced into 1980s. And then comes second generation markers in 1990s, SSRs, AFLPs, and their modifications. And currently the third generation markers are, you can classify, SNPs and ESTs, expressed sequence tags and single nucleotide polymorphisms. And another classification is based on distinction method. It can be either hybridization based markers. Hybridization based markers are, here your probe is hybridizing with your genomic DNA, and then you are assaying. So that types of marker system is called hybridization based markers. PCR based markers are, you're amplifying your DNA using the polymerization reaction, and then you are analyzing. Then you're going for analyzing. So those types of markers are called PCR based markers. Another classification is based on the location and functional significance of markers. Now the
So
markers
can you
can be,
show the first slide in the classification of molecular markers? Sir?
hello?
Can you show the first slide in the classification of molecular markers? Sir?
This one,
Oh, this one.
I need to
Okay,
explain.
sir. No, sir, no, sir. Thank you, sir.
Okay. Okay, shall I proceed or?
Yes, sir.
Yeah, okay. So yeah, based on location and functional significance of markers. Suppose if your marker is random, okay, you have a repetitive sequence, and based on that you are designing your marker, and this repetitive sequence can be bind at any part of the genome, right? So such kind of markers are called random markers. And linked markers, suppose you have a gene and you have mapped your molecular marker near to that gene, and since this marker and gene are in linkage, they will be segregating together. Okay, such type of markers are called linked markers. Now if your molecular marker is derived within the gene, the polymorphism, you are developing a marker using the polymorphism within the gene. Such markers are called gene based markers. And functional markers are, if you are developing a marker using the causative polymorphism. Suppose you have a gene, okay? A gene has two alleles. One is resistant allele, another one susceptible alleles. So the genomic part, the sequence part which causes that resistance, maybe it is a deletion or addition. So in such cases, in such parts, you can use, if your marker is developed from that causative polymorphism, that is called causative polymorphism. A deletion which causes resistance, or an addition which causes resistance. So those part, your marker is developed from those part, such markers are called functional markers. So in a crop breeding, the most utility is through functional markers. And yeah, so I will be explaining all this marker system in a very brief. The idea is to not to jam you with lot of information rather than get an overall idea. And this was RFL, restriction fragment length polymorphism. So even the name itself, it's very self-explanatory. Name itself explaining how the marker functions. Now what we do here, we take the genomic DNA, then we digest them with some restriction enzymes and make them into small parts. Okay. Then using a gel electrophoresis, we will size separate them. And then once the size has been arranged based on their size, then what we do that you will plot them these fragments on a membrane, nitrocellulose membrane in case of RFLP. Then we will label with a specific DNA probe. Okay, you will have a specific DNA probe of your interest to which you are searching in the genome, and you will tag them with a radioactive probe. Now you can visualize these probes in a X-ray film. So the banding, using the banding pattern, you can identify the genetic makeup of a particular individual or the trait. Now these markers are codominant, meaning you can identify between homozygous from the heterozygotes. Okay, both the heterozygote can be identified from the both homozygotes. And another one is next marker system is random amplified polymorphic DNA, RAPD. So RAPD markers are basically a short random primers. Okay, so you will use a single short random primers for as a primers for amplification of DNA fragments. And these markers, when they bind, for example, here, if you see here, when they bind the same marker will bind upstream and downstream in a closer vicinity, in an amplifiable distance, then you will get bands. And these bands are basically, since whether if you are if you are these short primers are binding, then you can call it as present. Whether you are getting band, then present. If there is no band, absent. So this is called dominant marker. And the advantage here is, here you don't require prior sequence information about the template genome, and it can be used to construct high density maps in several crop species. But the problem is these kind of markers are not reproducible. Maybe at one point you may get result, maybe in the next day if you try it again, you may not be getting the same result again. So again, these markers, RFLP and random RAPDs are not commonly in use these days. But yeah, simple sequence repeats, these are widely used markers these days. And although we are now turning to SNP markers, but SSRs are still relevant, especially in those labs where you don't have much funds, right? Now, if you see, the basis of the polymorphism is when the replication occurs, what happens is sometimes replication slippage occurs. Okay, due to this replication slippage, the repeats will be created. Now, if you see here, in parent one, so one allele have 5 repeat and another allele have 7 repeat. But in parent two, so these repeats are basically a short tandem repeats, like it will have repeats of 5 base pair. So if we have here 5 repeat, so the length of the repeat is 5 nucleotide or 5 base pair. So you'll have 25. The sequence length will be 25. Here it will be 5 into 7, 35. And then in the progenies, you can see that it will be, you will find in a different combinations of these repeats. And when you genotype, based on the length of the repeat, you can easily genotype the plant. These are codominant markers. And this is the basis of the basis of the mechanism of the simple sequence repeat. And you are actually amplifying that repeat sequence using the primers will be deriving from a unique sequences flagging these repeats, and you are basically amplifying the repeats. So these are all very having high utility in crop breeding to identify your trait of interest or your gene of interest, and they are codominant and highly reproducible. And the next type of marker system is called single nucleotide polymorphism. So as you can see the image in the right side, there are, in here it is GC, and here is the individual one, it is here it is G, and here it is A. So it is a G to A polymorphism, right? That's how we define the polymorphism in SNPs. So variation in a single nucleotide at a specific position in DNA sequence is called single nucleotide polymorphism. And again, they are codominant. So they can be easily scored presence of one or two alleles at this locus, and they are very easy to develop. They are highly reproducible and highly transferable across related species, and they are present everywhere. Okay, SNP markers are present everywhere. Their availability is huge compared to any other marker system. Therefore, their utility also they can be widely adopted in any species. Now, how to genotype using SNP markers? So there are basically few systems. We have competitive allele specific PCRs. Competitive allele specific PCRs. We have it's basically your quencher and dye system. Okay, if you will have a particular probe or primer which will be tagged with a quencher and dye, and the dye will produce a fluorescence. Okay, so when the quencher is present, the dye will not produce any fluorescence. So when the dye will be, that particular probe will be binding with your genomic region, then what happens is the quencher will dissociate from the dye, Therefore, it will produce certain signal. Using that signal, you can identify whether it is present which G or C or T or A is present there. Okay, so each four of these nucleotides will have different signals. Now, high resolution melting analysis is based on suppose you have this double stranded DNA, and this double stranded DNA, you have a dye, okay, like cyber green, which is binding specifically to double stranded DNA. So, and what you do is that you do a PCR reaction with this particular dye incorporated, and then at the end of the PCR reaction, you will apply heat and dissociate the double strands. So what happens is your strands will be separating into single strands. Therefore, the dye fluorescence of the dye will decrease. So this is called melting. And seeing this melting profile, you can decipher whether which SNP is present in particular location. And another widely used SNP marker system is SNP microarrays. These days we use very widely. You must have heard about 80K chip, 90K chip. In rice, we have 80K chip, 5K chip, 90K chip. So what happens here is that one chip having 80,000 markers. So at a time, you can genotype your individuals with 80,000 markers at a time. So it has got wider application. And another allele specific PCR. So for example, here you see, here we have three components. The first component one is allele one specific forward primer. Suppose you have two alleles, okay, one mutant allele and one wild type allele. Or suppose in this case you see you have two alleles. Here it is C, which is in purple, and here it is in T, which is in brown color. Okay, so individual one will have C and another individual will have T allele. So you need to identify those individual which having C. Also, you need to identify those individual having T. So what you do is like you will develop a primer in such a way that the primer is ending with your SNP of interest, SNP of interest. And the similar case, this is called allele 1. Here in allele 2, again the allele 2 is in the ending with this particular allele 2 forward primer is ending with the particular SNP of your interest. Now the thing is like even this SNP and this this is your allele wild type. For example, this is a wild type primer and this is your mutant primer, mutant specific primer. And maybe the wild type primer can also, there is only one mismatch, right? Only one, here there is only one difference between wild type and mutant is only this C and T, okay? So what do you do that? You will introduce a mismatch, okay? So with a mismatch, when there is two mismatch, it is not going to bind in untargeted locations. So this way you will develop, you will design two primers, and also there will be a common reverse primer. So what happens here is, the C and T, then when you do a PCR reaction, when you do allele 1 specific PCR with allele 1 specific forward primer and a common reverse primer, and here it will amplify only those regions which having wild type allele. Here you see wild type allele is, here there is an amplification, but here there is no amplification. But wherever wild type allele is present, they have amplified. But in wherever mutant allele is present, this is mutant, and these are its progenies, crosses progenies. So the first line is wild type, this is mutant and its progenies. So mutant has not amplified. Now mutant specific primer if you use, so the allele 2 specific primer, otherwise, or mutant specific primer, if you use it, here you see that in wild type it has not amplified, but in mutant it has been amplified. And those individual which carrying mutant allele, it has got amplified. Now, if you decipher, you will find that, if you decipher their genotypic interest, you can, genotype, you can find that this is capital A, since both of these you adjoin these two PCR result and you can decipher the genotype of the individual, right? Here it will be capital A, here it will be small a, again capital A, capital A. Here since both wild type and mutant allele is there, it will be your capital A, small a, heterozygote. So this is how you genotype, right? Now, okay. Okay, I think I'm not able to. Am I audible? Hello?
Yes, sir, you're audible.
Yes, sir.
Yeah. Just a minute. I have a difficulty to changing my slides. It's not moving. What I will do is like I will reshare it. Can you see my slides in full screen?
Yes, sir.
Yes, sir.
Okay. Yeah, so another marker system is called genotyping by sequencing or GPS. So this marker system is mostly more relevant in terms of in the crops where the reference sequence is not available. Okay. So what here you can do is you can take your genome, isolate your DNA, digest it with restriction enzymes, making into small fragments, and to get the identity of these small fragments, you will bind them with a specific adapters, and these adapters ligate with these specific adapters, and adapters will have certain barcode. So with that, you can identify what, which sequence it is, like which you can give an identity to the fragments. Now you will pull all these ligated fragments, then you perform a PCR reaction. And so once it amplified, then you can go sequencing with high throughput sequencing. And once the sequencing has been done, then you can go for a SNP calling using bioinformatics tools. Then you will get the genotype of those particular individual. So genotyping by sequencing has got a very wide application, especially in crops where already reference sequencing has been not done or high quality reference genome is not present. So those cases, it is very widely useful, especially has got wider application in genome analysis, genome association studies, and even genomic selection. So so with this you will have an idea about the markers. Now we will see how these markers can be used for a marker-assisted selection in crop breeding program. Now, if you recall our, the previous slide, the first few slides, the same plants here, right? These plants, they are in their seedling stage, in a very early stage. You sown them in the field, and the plants have been emerged. And what you have to do is you have to isolate the DNA, okay? And once you isolate the DNA, go for PCR reaction. You will have certain genes governing the disease, and there will be certain molecular markers linked or associated with this particular gene. So you can amplify those particular markers, and then you perform a PCR reaction for amplifying those particular markers or gene. Now what happens is that then you do a gel electrophoresis to identify the genotype of each plant, and you will have a fair idea about which of these plants carrying the gene for resistance and which of the plants carrying not. So you can easily identify, your plants you have to get selected. Okay. So you can do the advantages here. You can do it in very early stage. You don't have to wait till the disease occur, till the plant get maturity, and you don't have to worry about the environment for its correct expression of the disease. Here, your selection will be much accurate. You are not relying on any external factors. You are only relying on its gene and its genotypic content, right? So you will have a better chance for selection here. So this is the a broader application of marker assisted selection. You'll have different, again, using markers, there are different specific applications. So since the audience is very wide, I don't I don't want to be, go into so technical details about all those things. And when we discuss about the molecular markers, certain applications are, you can do fingerprinting for particular varieties or strains for its identification. And the molecular markers will give a marker profile that will be very unique to each of these varieties. Using that, you can identify, okay, this variety is this and this variety is that, right? And you can map your genes, quantitative traits. Also, as we discussed, will have an efficient marker-assisted selection can be performed using the markers associated with the title linked gene or quantitative trait locus, loci. Now the markers will help you to clone your gene of interest. And also you can establish phylogenetic relationships, you can perform diversity studies, and you can select the suitable parents for your hybridization program. Also, you can identify, the pathogen races and its biotypes, and it has got applications in heterosis. Also, maybe not one gene is important every time, right? A better plant is an output of so many better gene combinations. So you can pyramid better genes, better traits using molecular markers, right? So this was the era of molecular breeding. Now we are shifting towards genomic assisted breeding era. So genomic, we have, due to the advance of developments in genomics and sequencing, new sequencing technologies, low cost sequencing technologies. So getting a sequence information become more cheap. Okay. So genomic selection in this era, the genomic selection has a very huge role. So I would like to explain very brief about what is genomic selection. Okay. And what its application in crop breeding. So genomic selection, okay, so this is equation called Breeder's equation, okay? And this equation is given by R.A. Fisher. Later on, Dr. Lesh modified this equation. So I will just explain what is this equation means. So I think I don't have to explain about what is genetic gain, right? Genetic gain is what? From each generation, how much yield improvement or performance improvement for each trait has been happened. That is called the gain. So that is called the genetic gain. Now the genetic gain has been, it has been controlled by several factors. The first one is selection intensity, and another one is prediction accuracy, and another factor is genetic variation. So if you can increase any of these three factors, you can increase the genetic gain. And L is called generation interval, and meaning the lifespan of a crop, okay, lifespan of a generation. If you can decrease the generation interval, if you can, for example, you are growing three seasons, you are growing 3 seasons in a year, or 3 crops in a year, if you can increase into 4 or 5 crops, you can increase the genetic gain, okay. And inbreeding depression is what happens in cross-pollinated crops. So it's not applicable to self-pollinated crops. So in cross-pollinated crops, when you self, you will have inbreeding depression. So if you can decrease inbreeding depression in cross-pollinated crops, it also augments the genetic gain. Now genomic selection. So genomic selection basically, okay, in marker-assisted selection, what we were doing, we were only selecting few markers. We have few genes. For example, yield, we will have 2 or 3 genes we will be focusing on, and using those genes we used to select. But there are two types of traits. One is qualitative trait and quantitative traits. So in quantitative trait that are affected by several genes, they are polygenic, okay? So not just one gene, they are affected by so many several genes. That's the so it's very difficult to manipulate each gene. So that's the difficulty of you will face to improve quantitative traits. Now, genomic selection focus on, instead of focusing on 1 or 2 genes, here you assume that you will, for all those markers you will take into consideration. Okay, if you have 10,000 markers distributed all over the gene, you assume that all these 10,000 markers will have some role in your trait of interest. and you will use all these 10,000 markers to predict the breeding value. Breeding value is what? The performance of an individual for a particular trait. Okay. Now you will estimate the breeding value using genomic data. That is called the genomic estimated breeding value to select your favorable individuals. And this genomic selection can increase selection intensity. So that we will be explaining again in, okay, I will explain the same slide. And how the genomics, okay, then selection intensity is what? For example, you have 1,000 individuals, okay, and you are going for a 10% selection. Then your selection intensity will be what? Yeah, you will selecting 100 plants. It meaning 10% selection. And if you are selecting 1%, if you go for more intense selection, you can go again select 1%. So in 1,000 plant, if you select 10 plants, it will be 1% of the selection. It means that you are going for more selection intensity. But if you selecting 100 plants, it will be comparatively lesser selection intensity. You will get at least 100 plants. So if you increase selection intensity, according to this equation, you can increase the genetic gain also. So the fundamentals of the genomic selection says, through genomic selection, you can increase the selection intensity by from a population of large number of plants, you can select very few plants, even though by retaining a considerable amount of plants, but with higher selection intensity. So this can lead to more rapid genetic gain. So this is the fundamentals of the genomic selection. So genomic selection can increase the, since you are considering all the alleles or all the genes present across the genome. So here you will have a better chance for select, your accuracy should be, instead of selecting one or two genes, you are selecting, here you are going with the whole genome for the selection. So with this, you can have a better selection accuracy. And so if you see how the genomic selection can be employed in a crop breeding program, suppose you have a population here of 10,000 plants, okay? What you do that you divide the population into two randomly. You will have a breeding population and you will have a training population. Now, what you have to do is this training population, at least suppose you take around 500 plants, okay? So 500 plants are 500 genotypes. This 500 genotype, you will phenotype them. Also, you will genotype them. Then you will create a model. It's called a genomic selection model. So you will associate your phenotypic, for example, your phenotypic value with your genotypic value. And whatever the information you are getting from genotype, you will take as a cofactor. And with that, you will create a model. Suppose if particular genomic content is there, then it may produce this much of phenotype. In terms of yield, if we say, if particular, if this is the genetic constitution of that particular plant, it will produce 10 gram of yield. If this is, if the genetic constitution is different, it may produce 20 gram of yield. So the model works in that way. Okay, Y is your phenotypic value, and other things are your cofactors. Here you will use your genotype value as one of the cofactors, or genomic information as one of the cofactor, and with this you can predict your genomic estimated breeding value. Okay, so now what will happen that since you will validate this model, if you feel that this model has a considerable selection accuracy, you will predict raised to how many? If you have 5,000 plants and you have here 500 plants, you have rest of 4,500 plants. You will, instead of growing them in the field, what you have to do is that you grow those, you will isolate the DNA of those 4,500 plants. You will genotype them, and with that you will predict uh, the genomic estimated breeding value of this 4,500 plants without growing them in the field. So it's you need a large number of area to grow 4,500 genotype in a field. It means that 4,500 plots. So it requires of hectares and hectares of area, which is so resource consuming and practically very difficult to execute such kind of operations. So here, what you do is that you just genotype them. You have already one equation here. You will apply those equation and predict the genomic estimated breeding value of those 4,500 plants. Now you have GBVs of 5,000 plants. Now you can go for, now you can go for very intense selection. So this intense selection will you allow you to, you can increase your selection intensity. For example, you have, suppose in a field practically you will be able to raise 100 plots. So if you do 10% of the selection, it means that you are selecting just 10 plants, and you are only left with the 10 plants, and that is the maximum selection you can do. That is 10 percentage. But 5,000 plants, if you do for 1% of the selection, even how much it will come? It will come around 50 plants, right? Even with 1%, you can select 50 plants. So here you can increase your selection intensity. So you must have heard about speed breeding, right? So if you can increase the number of generation per year using speed breeding is nothing but you are here, you are manipulating the environmental condition like light, intensity of the light, wavelength, and the temperature, humidity. So you are controlling all those external factors so that the plant will mature in very short time, so that uh, the their generation or lifespan or it will be reduced, and you can carry out more number of cropping season, or you can, yeah, more number of generation per year you can, you can have. So with this, again, you can use the genomic selection. And if your prediction accuracy is high, again, this genetic gain will be higher. So these are the ways which genomic selection can be employed, and the markers can be much useful for uh, increasing the genetic gain. So another concept I would like to introduce you all, haplotype based breeding. And haplotypes are basically specific combinations of alleles at multiple loci located on the same chromosome that are inherited together. So if you see this, you have here 3 individuals. Okay, it's all, this is from a same gene. You have 3 individuals. So within these 3 individuals, you find that there are a lot of SNPs are present. Okay, and also, so this combination, if you see, SNP means, so these are monomorphic nucleotides. Okay, you monomorphic bases which you will not be considering. The polymorphic bases where you have polymorphism, those are called SNPs, right? Which I have explained previously. So if you include all these SNPs, you will have different combination like TG, TCT. From a same genomic region, you will have multiple combinations. And most importantly, from each generation to generation, these combinations or haplotypes, they inherit as it is without any recombinations. And now, when you evaluate, for example, in this case, we have 5 haplotypes. Okay. Now you check the phenotypic performance of the 5 haplotypes. You find that these haplotypes are, so haplotype 1, it is giving a different yield. Haplotype, this particular haplotype 2, this is giving the highest plant weight. Okay. But the lowest plant weight is giving by the haplotype H5, right? Now you have to select the haplotype 2 for getting the better result, right? So this is called superior haplotype. So this haplotype information can really accelerate the crop breeding programs. So I will, we will give you a very graphical outline of how it happens in a real crop breeding program. Suppose you have a germplasm. Germplasms are group of genotypes. Okay, you genotype them, you will phenotype them. Okay, and for suppose this is a disease resistance, you are performing this. What you go for is a mapping study. You will map. Again, all these are all, if you go into details, we may not be able to end in within not even 1 hour. It may take, a lot of time. So I'm not going into much details. So we will perform a genome weight association study. And with this, we will find out the genomic regions which are associated with this particular trait. Okay, what are the resistant genes, right? Once you identify your gene of interest, so you basically will locate your MTA which are in LD with this particular gene, any particular gene, and Since if the then what you have to do is that using the bioinformatics tools, you will identify the haplotypes present in that particular gene. So once you identify these haplotypes, you will perform a haplopheno analysis through which you will identify superior haplotypes, which I have shown you in previous slide. This haplotype which is giving you best performance. Then, so you will identify the plant which having the superior haplotypes. Next, what you have to do is that you cross this plant with an inferior haplotype, and you will transfer this superior haplotype into your better into another plant, and thereby you will create a superior haplotype with a better stress tolerant variety with a superior haplotype. So this is how happens in a crop breeding program, how they develop superior varieties. So haplotypes, again, it has got a huge application. I just outline how it happens in a very broader perspective. So with this, I would like to end my presentation. I hope maybe I have used too many technical words and jargons, but I'm happy that if some of you got some benefit out of it. So thank you so much for this opportunity, and I would like to acknowledge my sincere thanks to the Varde College and all the organizers for this program. And it's been a great pleasure to meet all of you. Thank you. So if you have any questions, yeah, we'll discuss it now.
Yes, sir. Yes, sir. Am I audible?
Yeah.
Okay. So thank you so much for your informative session. It was really wonderful. So we'll just take a few questions. Let me just check from the chat box if there are any questions. Participants, if you have any questions, please do ask to the sir. Till the meantime, I'll take questions from my side.
Yeah, sure.
Sir, there is one question which has been asked.
Okay.
The person is asking ki how the marker works.
Okay. How the marker works. Okay. So I think I was explaining how the marker works throughout my presentation. I don't know where the miscommunication has happened. So the question is not to be clear. I mean, can you just clarify more about the question? Probably that is half of the question.
Mr. Manoj, could you please ask the question to sir?
Hello, sir.
Hi, hello.
Good afternoon, sir.
Yeah, good afternoon.
Sir, in where, sir, that markers are used?
Okay, great. So, yeah, as I said, for example, okay, I will explain it again. Suppose you have a, yeah, I will tell you, for example, maybe not in plants, I will tell you in a human example, right? Suppose a person, we have a group of people, okay? Suppose some kind of disease is very common in that population, okay? So we cannot wait the person to get the disease, and we can say that, okay, this particular guy has this disease, right? Some of the diseases are genetically, you may be knowing that some of the diseases have a potential to inherit through genetics, right? The genetics affect the genetics can bring the disease to people. For example, diabetics, okay, it has a genetic basis. If your parents have diabetes, you are likely to have diabetes too. So suppose in some of your suppose some of your in a population where what basically we have to do is that we will genotype, and if we have a marker which are associated with the genes of this disease, with genes which control, which genes govern this disease, what we do is that we don't have to wait till the person get disease. We can predict it very early so that we can take precautions, right? So we will isolate the DNA of each of the individual persons, and then we will check using the markers. We will check whether the gene which caused this disease is present or not. If yes, we can give that result. So that is one of your application which maybe you will be able to understand it very clearly. So in plants, so similar way, for example, the rice, maybe the rice or wheat which you having today, they are not easy to grow. Okay, so if you grow a plant in a field, there are a lot of factors which trouble farmers. Okay, one is your, if there is a water shortage, the plant may die. If there is a disease infection happen, the plant may die. So the farmer undergoing all these struggles. Okay, so a breeder, what a breeder will do, a breeder think in this way that farmer do not have to endure all these difficulties. Instead that, what if we can provide a variety, or variety is what I am referring to the plant. Okay, a variety which can be withstand disease, a variety which can withstand drought or salinity or any difficulties, any abiotic stresses or biotic stresses, so that farmer do not have any problem. Okay, farmer do not have to apply pesticide. So for this, there are genes which governs all of these factors. Okay, there are genes which governs, there are genes which gives resistance to these plants against any diseases or insects. There are genes which governs the plants. If the gene is present, the plant can withstand in any environment. Plant can, if the gene for drought tolerance is present, in a drought condition, you can grow the crops. You will get better yield. So how you know that whether the gene is present in this plant or not, right? There the marker comes. So what we do is that once we map, first we have to map the marker, the lot of markers with your gene of interest. And once you find that, okay, this particular gene, this particular marker is associated with this particular gene, then these markers, you will just screen your population for you, so in search of that particular marker, a specific marker. And if you have that particular marker, then you can safely say that, okay, then the gene which I am searching for is present in that particular plant, so that I can select those plant, and with that I can develop a variety, so that it will be, that variety can help a farmer to grow those plants in a drought condition or any difficulty condition. I hope it's clear your doubt.
Yes, sir, I get it. Thank you, sir. Thank you for your explanation.
Right.
The next question is from Mr. Prathamesh. He is asking, can you explain more about the third generation markers?
Yeah, third generation markers are basically, okay, don't get it confused with third generation sequencing. So third generation markers are just based on the chronology. It's there are basically ESTs and SNPs. So I think I have explained you well about what is SNPs. ESTs are expressed sequence tags. So how you get this EST information is, for example, it is from mRNAs. Okay, expressed means mRNA, right? So this mRNA, from mRNA you will you can get the information of the complementary DNA, cDNA. So with using that information, you will design a marker. So this marker will target only those genes. Okay, if I say a crop genome, a plant genome, a genome has two components: coding sequences and non-coding sequences. So if your marker is designed from non-coding sequences, it may not have much utility. Okay, but expressed in the sequence tags are especially designed from the coding sequences. So coding sequences only can give to mRNA, right? So those expressed sequence tags will have utilize the information from those sequences, and you will develop the marker. That is ESTs. And yeah, so now the third generation marker, the advantage is that especially SNP markers are very high throughput. So you can, at a time, you can genotype thousands of individuals using thousands of markers. Right. Yeah, I think
There is one more question.
Yeah, sure.
Mr. Sal is asking, please explain the DF, that is inbreeding depression in breeders equation.
Okay. Inbreeding depression. Okay. The importance of inbreeding depression is suppose it has application even in humans also. Inbreeding depression is what, for example, a cross-pollinated species, meaning that the species have more heterozygous genomic content. And a cross-pollinated species, when you self them, okay, okay, I will explain again in a human perspective that you will have to understand it. Okay. Uh, okay, maybe. Yeah, I can. I explain in a plant basis. Okay. So if the what will happen when you self, the deleterious alleles will be expressing in that particular individual. Okay. For example, there are maybe, for example, in a heterozygous condition, capital A and small a. Suppose capital A is your favorable allele and small a is your non-favorable allele. Okay. So if capital A and small a is there, always capital A will express, right? That's what Mendel says. So what happens is when capital A is there, it will safeguard you from small a, from expression of small a. Okay. But when what happens when you self capital A small a, you have three possibilities. You will get small a, you will get capital A small a, and you will get capital A capital A. So the individual which having small a, okay, the recessive homozygote, there is a more chance of presence of deleterious alleles. So these deleterious alleles are expressing there, so that will affect the performance of a particular individual. So that is called inbreeding depression. So it happens mostly the genomes, the plants or crops which are having heterozygous in nature, they are cross-pollinated. So if you can minimize the inbreeding depression, again, you can increase the genetic gain. That's the importance of genetic gain in the breeder's equation. That's the importance of inbreeding depression in the breeder's equation. And even in humans, we say that, we should not marry the relatives, or that is again related with inbreeding depression, right? Yeah, I hope you understood this.
Yes, sir. Thank you.
Right.
Yes, sir. There is one more question which is asked. Can you please explain that if there is no genomic information available for a particular crop, that is, if a plant gets infected with a biotic or abiotic disease, then how we can identify the genes associated with that environment cue?
Oh, very good question. Yeah. So suppose you have, okay, you don't need any all the time, but you need to know, you need to have enough genomic information so that you can develop markers, right? Suppose you have marker information, okay, and the marker information, the which marker present at what part of the genome, you have that information, and what you have to do is like you have to do a mapping. Okay, you have, suppose you have 100 marker or 200 markers. So, and the physical position of these markers also, this much is required at least, right? And the first step is, even if you don't have, first step is to develop those markers. Okay, so once you have these markers, what you have to do is you have to create a mapping population. A mapping population means, suppose how you create is like you have a plant which is two plants. Okay, one plant having disease resistance, one plant is showing disease susceptibility. So you cross two plants, you create F1, and from then again you create F2 population. Okay, I'm telling the simplest mapping way. Now you know the F2 population, how the segregation of the markers occurs. It is 1:2:1 for a single marker, right? Now what you do is you will genotype with, you have 100 markers, as I said, you will genotype these 100 markers for this. For example, you have 200 individual in F2 population. So you will genotype all these 200 individual with the 100 markers. Also, you will evaluate this 200 individual for disease resistance. So you will subject the disease on those 200 individuals and you will evaluate them, which individual having disease resistant and which is individual having susceptibility. Now this is called phenotyping. So now this phenotypic data you will associate it with your genotypic data. That is your marker information. From there, you will be able to closely, using that statistical tools, you can associate. It's called QTL mapping. In F2, it's called symbol mapping. And if you can If in other population, it can be either QTL mapping or even association mapping. So different mapping methods are there. So proper using the proper statistical tools, it is nothing but a regression study, right? Regression analysis, how you do it. That's what mapping is. Basic, very basic. So using the proper statistical tools, you can associate which marker is associated with your trait of interest, right? This is how you identify your uh, gene of interest. From there, you have to clone. Okay, that's the first step. Now you identify markers. Now you have to map it. Now you have to fine map it. After fine mapping, you identify very narrow genomic region which governing this particular trait. Now you have to clone it, and the cloning, then you have to sequence it. So that's how it works. You identify the proper genes. That is one way. You can do in other ways also. Yeah, I hope it clear your doubt.
Yes, sir.
Hello.
I guess that's all the questions from the audience side.
Okay.
Hello, sir.
Hi.
Hello, sir.
Yeah. Please go ahead.
Sir. Good afternoon, sir.
Yeah, good afternoon.
Sir, basically, if we want to identify our gene of interest, right, with the associated marker. So, in first place, how we are documenting that a marker is associated with that gene of interest? Means for reference, means how we are get to know that marker is basically related to that gene only.
Yeah. So, yeah, the concept is okay. The concept here is, for example, can you see my pen? Oh, sorry,
Yes, sir.
No,
yeah,
sir.
yeah.
No, sir, no.
Oh, okay, fine, leave it. So, for example, okay, the concept here is your gene is your marker is you must have heard my previous answer, right? The question which I have answered for how mapping has been done.
Yeah, sir, I heard.
You heard that. Okay. So normally after mapping, you will identify the markers which are present in the close vicinity of your gene of interest. In a physically, they will be very physically proximal to your gene of interest. It means that nearby your marker, there is a gene. So it's called linked markers.
Okay, sir. Means,
Right.
but
So
how?
how you make sure is that you have to map it first, then you have to validate it. So after mapping studies, you will identify the markers and you will validate it. Then you will make sure that your marker is in close vicinity with your gene of interest. Either you can do this through QTL mapping or genome weight association studies.
Okay, means, sir, in on NCBI website, we can find that data, right? Marcus data.
NCBI website marker data, I'm not sure able to find in NCBI website, but there are different databases these days for each crop. In case of rice, there is called a RABDB, a rice annotation project, and in case of maize, again maize database, maize genome database. So each crop wise, you can find it. Maybe not in a single place you will find all the information. There is another tool website called Grameen. In Grameen, all the plants which belong to Graminae family, they have marker information and their database is present. And it's really easy these days. You can just have to search in the literature search. Okay, go to Google Scholar, just type your trait and the information you want. You will find a lot of literature, and that way also you'll find out these informations. Even NCBA also having a lot of information. Mostly it's a genomic information, gene and its sequences. If you want to find any gene and its sequences, ESTs, and mRNA and its sequences, all this information you can find in NCBA. Yeah.
Yes, yes, sir.
Okay.
Sir, yeah. So nowadays, which technique we are using for breeding techniques means for using molecular breeding, which is the latest technique, or we are looking to apply in future also for breeding purpose.
See, it depends on the purpose again, right? Breeding is it has in various objectives, right? So we have, like I explained about genomic selection. Genomic selection is again, these are all selection tools. Okay, the idea is you have to develop a better variety. For that better variety, you have lot of plants from which you have to conduct selection. So whatever you can do is selection tools. Now coming the genome editing has a huge potential now. So genome editing, you can modify the trait of interest as you want. For example, you have a gene for susceptibility. You can modify, tweak the gene and make it resistant. So the same plant will be resistant. So genome editing has a huge potential, and I have explained about haplotype based breeding and genomic selection and how it. But yeah, all these are selection tools. Ultimately, a breeder has to grow his varieties in his field. Even if you have all these tools, a, a better variety which will reach to farmers will be only those variety which can withstand to any environmental stress, and with all this environmental stress and the input conditions, they should give better yield, right? They should, then only the variety will be successful. So for that you need to conduct field trials. So the basic selection process all remain same, but again the novel genetic tools can accelerate the breeding process. Maybe the things which would have taken for years to come, you can do it in very few months. Maybe you can cut short a few generations of selection cycle. So yeah.
Sir, nowadays we are using CRISPR also. So I want to know the use of CRISPR in plants and all in humans. What are your the view on the CRISPR? Is it safe or not? Like,
Yeah, it's a good question. So, okay, so initially we have the concept of transgenics, right? So transgenic is about, you are transferring genetic content of a particular gene from outside the outside the genus. That's called transgenic. And the problem is like from bacteria, okay, from your, if you take any bt cotton, you must have heard about it. So here you are taking the genes for insect resistance from a bacteria called bacillus thuringiensis. Using its mechanism, it will provide an endotoxin, right? So the problem in this case is like, this is not a naturally available genetic variation. Here you are going, you know from an outside the taxa, you are bringing those sequences, you are merging those sequences into another species or another genus. So this has been actually led to a lot of outcries and protest, ethic reasons. So now the genome editing, what you are doing is like, you are not doing any of those things. You are here, uh, what you exactly doing is like, you are manipulating a particular gene. You are not adding any extra elements, not any extra sequences. You are just tweaking. These changes are same as which could have been brought by conventional breeding, same change, but you are targeting a specific gene and you can editing that particular gene, okay, which is a very novel technology. In a conventional breeding, it is very difficult. We cannot target a particular gene, okay, not even a big genomic region also. You can only select the plants, or you can even if you do mutations, you don't know where the mutation lands, okay. So it is basically a mutation, but here what you are doing, mutation is allowed in the plants, right? We are doing, there are a lot of mutant varieties which we are, which were in the cultivation. There was no protest. Same time, this is kind of a genetically induced, specific mutation using biotechnological tools. That's all. So genome edited plants are very much safe. Okay, you don't have to worry about its safety at all. You are not doing any variation which is bringing outside of the genus. outside of the genus genome. And that's the reason why in India, genome edited crops are not regulated. You must have, I think the last year that notification has come. So now there will not be any regulation for genome edited crops in India. But unlike in transgenics used to have years and years to get approval. So far in India, only BT cotton was only the crop which was, since it's a non-consumable, it's a cash crop, they allowed it. But after that, I don't think, it was very difficult to get approval from GAC. Now with the latest regulation requirement, genome edited crops are free from all these regulations and very safe to consume.
Okay. Yes, sir. Thank you so much, sir. So rest of the particular doubts, if anyone has, sir will put his email ID. You can contact through his email ID if you have any doubts. Sir, we'll move on to the you can vote of thanks.
Sure.
Okay, so I, Mr. Pushpa Bojne, on behalf of Patkar-Varde College and the entire faculty of Department of Biotechnology and Microbiology, consider a great honor to propose a vote of thanks. First of all, I would like to propose a sincere vote of thanks to our principal ma'am, Dr. Pratibha Gaikwad, for gracing the national webinar on Biofacet, Sustainable Agriculture Through Plant Breeding, with her eminent presence. Thank you, madam, for the interesting and motivating speech, which gave a beautiful opening to this webinar. I will also like to extend a very hearty vote of thanks to our speaker, Dr. Abhijit KP sir, for taking out precious time from his busy schedule to enlighten the students as well as the faculties and making this webinar a great success. I will also like to express a profound gratitude to our CEO, Dr. Mala Khaka ma'am, Chief Coordinator, Mrs. Suchita Dalvi ma'am, for their moral support and guidance. We are also grateful to our sponsoring organization, RUSA. This event wouldn't have been successful without the constant support from our staff members and wouldn't have run smoothly without our technical team as well. Last but not the least, thanking the wonderful participants who have turned up in such a great number from all over the country and for their active participation throughout the webinar. Once again, thanks to all for their cordial cooperation. The feedback link for the participants will be posted in the chat box. The participants are requested to fill on priority basis, as the link for the feedback will be open for the next 30 minutes only. The respective participants will receive the e-certificates after 1 week. Thank you once again.